20 feet long. The mature sperm in the epididymis will pass out of the man's body through ejaculation 200 to 300 million at a time. Or they will eventually die and be reabsorbed. These sperm carry the man's genetic material and they are fully mature. But they are not yet able to fertilize a woman's egg. They do not attain that ability until they are actually far up inside the woman's body. They may stay in the epididymis, the bag-like structure on the left, for several weeks. While here, they develop the ability to swim. A sperm propels itself by means of a long tail, a flagellum, which lashes vigorously from side to side. This extraordinary activity has but a single purpose: to deliver the genetic material in the sperm's head quickly and precisely to the woman's egg. On the tip of the sperm's head is a layer of enzymes and enzyme inhibitors. The inhibitors preserve the enzymes, which will be needed later to penetrate the surface of the egg. This is how the sperm's head looks without its enzyme cap. The material of the head itself is loaded with the actual genetic information which will be transmitted to the egg. These are the strands of DNA which will be transformed into chromosomes. This is one of the many fuel packets arranged along the midpiece of the sperm. The fuel consists of sugars, which is converted into energy for the sperm's locomotion. The propulsion system of the sperm's tail is made up of a bundle of cords or filaments. The filaments are covered with thousands of tiny hooks. As the hooks intermesh, the whole bundle begins to bend, and the tails of the sperm begin to whip around like this. It is a primitive but highly efficient system of propulsion. The system does not function perfectly every time. On the average, up to 20% of a normal man's sperm are deformed or imperfect in some way. Perhaps one reason why a man produces so very many sperm is that so very many things can go wrong with the sperm themselves. They may have 2 tails, like this one. Some even have 3 tails. In another common deformity, the heads of these sperm appear to be nearly severed from their tails. This sperm has been attacked by bacteria. And this one may have been produced too rapidly. It will never complete its development. White blood corpuscles, the large round cells, are often found together with defective sperm. Their presence indicates the likelihood of infection, fever, or even a common cold. Most of these sperm deformities could have been caused by just a slight elevation in the temperature of the testicles. Environmental conditions, too, often damage sperm production. Overcrowding, stress, smoking, chemical pollution of air and water, occupational hazards, radiation, poor nutrition, all part of modern life. Even our modern clothing may affect sperm production. Tight blue jeans, for example, hold the testicles up close to the body, perhaps raising their temperature those few critical degrees. But in spite of such obstacles, human beings have been successfully reproducing for millions of years. Since the first organisms swam in the sea, the cycle of life has been continuous, fueled by the need and the drive to reproduce. Among human beings, that drive is intimately linked with attraction and desire. Affection and romantic love may initiate the dance of courtship, which may lead to conception. Sexuality, affection, and tenderness can all be part of the complex rituals of mating. But desire and physical love possess a silent language all their own that signal partners' readiness to each other. The eyes are part of that silent language. Visual impressions stimulate and excite special nerve cells in the sexual areas throughout the body. The skin, too, is part of the silent language. It is amazingly sensitive and responsive to the touch. It contains millions of these sensory receptors, which, when stimulated, direct messages of sexual arousal to the brain. Many of these are concentrated in the body's most responsive areas, the erogenous zones. One of these, the man's penis, is densely packed with sensory receptors, here seen at enormous magnification. They relay messages of sexual stimulation to the brain, which triggers the complex sequence of events leading up to ejaculation. The length of the penis is made up of many small cavities called erectile tissue. When a man is sexually aroused, the signals from the brain cause these erectile tissues to become engorged with blood. As we can see in these images from the thermal camera, the sudden flow of blood to the penis raises its temperature. The man's blood pressure, heartbeat, and respiration increase. And the blood causes the penis to become erect and elongated so that sperm can be most easily delivered to the woman's egg. In the epididymis, the hundreds of millions of waiting sperm, which will soon be expelled from the man's body, accumulate in a mass. As sexual arousal reaches its peak, the entire male sexual system must function perfectly for ejaculation to take place. Cowper's gland, prostate, seminal vesicles, and epididymis. This is the actual journey made by the sperm. At the climax of sexual arousal, the man's nervous system triggers rapid involuntary contractions of the muscles in the walls of the epididymis. These propel the sperm up into the man's body along this tunnel-like tube, the vas deferens. The sperm themselves travel the distance, a little over 12 inches, in only a few seconds. The seminal vesicles release nourishing fructose, which blends with the sperm as they pass by. They mix with the prostate fluid. The blended semen approaches the urethra. Its total volume is actually about 1/2 a teaspoon. This is the last leg of the journey. The semen containing the sperm is forced out through the penis by the man's muscle contractions. And at last, it is ejaculated into the woman's vagina. As the semen enters the woman's body, it immediately slows down and coagulates. Perhaps it becomes thicker to ensure that most of the sperm stay within the vagina. Or perhaps the coagulation is a kind of defense, for the environment in the vagina is extremely acid. The acidity protects the woman against bacteria and infections, but it is also dangerously inhospitable to the sperm. Nearly 1/4 of them will die almost immediately. About 20 minutes after ejaculation, the semen becomes fluid again. The surviving sperm become vigorously active, swimming rapidly against the downward currents in the woman's body. There is great urgency, for the sperm will remain viable, able to fertilize for only 28 to 48 hours. This is the sound made by thousands of active sperm. The sperm's great activity has but one goal: to find and fertilize the egg. There are so many of them that success might seem assured. But the barriers, too, are numerous. The high percentage of defective sperm has already lowered the odds. So has the vagina's hostile acid environment. And the woman's own defense system attacks the sperm. They are unwelcome cells from another organism, and they are potential enemies. Her defense cells aggressively protect her from the invaders by destroying them. The sperm are programmed to seek the egg, but some of them try to fertilize the first round object they find. In this case, an ordinary body cell. Still, in spite of the obstacles, hundreds of thousands of sperm make their way up through the vagina to the cervix, the mouth of the uterus, which will lead to the fallopian tubes and the egg. The cervix secretes strands of a special fluid protein called mucin. During ovulation, it is very liquid and easy for the sperm to swim in. The strands of mucin provide tiny channels, less than 1/100th of a millimeter wide, directing the sperm closer to their objective. But many of them never find the strands of mucin, and they will die in the vagina. Those that survive will swim up the mucin channels toward the uterus. They swim in dense bunches. The outer ones may protect the inner ones from the woman's acidity and defense systems. Once inside the cervix, the sperm continue on their journey. Some remain here, perhaps to provide a backup for those that continue on. The rest proceed up through the cavity of the uterus in search of the egg. Their number is noticeably reduced. Here, from inside the uterus, we see the openings to the two fallopian tubes. The sperm swim toward them. One leads to the waiting egg, the other to an empty tube. Only half of the few remaining sperm will swim up the tube which holds the egg. The tubes are lined with millions of tiny cilia, the same cilia which helped draw the egg from the ovary toward the uterus at ovulation. These cilia sway constantly, creating a gentle, downward current that the sperm must swim against. Some sperm get stuck in among the cells lining the walls of the tube. Others lose their sense of direction. During this part of the sperm's journey, the layer of enzyme inhibitors at the tips of their heads is slowly being worn away. The sperm are fully capacitated and able to fertilize the egg, if they ever encounter it. Of the 200 million sperm that begin this long journey, only about 50 ever reach the egg. The egg is surrounded by two layers of the nutritive cells which have nourished it on its journey in the fallopian tube. The sperm immediately release their digestive enzymes to break through these protective nutritive layers in order to reach the egg itself. At best, only one will eventually enter and fertilize the egg. The joint force of their exertions starts the egg rolling around like a mysterious celestial body.